Hartwig and I go way back, and I think we met at a meeting in Switzerland, which was when Holly, I think, met you too.
Yes.
And everybody was. Nobody had met Hartwig before, and everybody was amazed with this guy who seemed to know everything. And at first we weren't sure, but in fact he really did know it. I think we're a fellow in Munich at that point, and everybody was very impressed. So that was in about 1998 or something like that. And since then, Hartwig has had a really stellar career and is now the boss of two very impressive and complex sounding institutions. One is the Danish Research Center for Magnetic Resonance, and the other is the Center for Functional and Diagnostic Imaging and Research, which are embedded in some complex way. They are in Copenhagen and are really turning out beautiful work. I think there are very few people around who think this rigorously and yet creatively about the potentials and problems in sound-based point stimulation. So rather than take up more of your time with more introduction, I'll let Hartwig speak.
Thank you very much for this kind of invitation. The last time I was at NIH, I was actually in Hartwig. That was several years ago when we were looking at the 7 Tesla scale.
I remember that.
Yeah. So I hope we will have a vibrant discussion today too, but not on the geophysical scale. So the title of my talk is Non-Invasive Transcranial Brain Stimulation, and for me it's really important that it's NTBS and not NEBS, because if you just call it non-invasive, speaking to you is non-invasive brain stimulation, and I think the really important part here is that the stimulation occurs through the skull. My disclosures are here, not many, and the funding is most of the funding I got in recent years was from the Multimodal Foundation called Biophysically Adjusted Orthotic Stimulation, and it's a synergy grant. I will start off with this problem we had in the field, and that was an editorial that Sibner and I wrote about the variability of plasticity-induced effects. I would say that the field is in a crisis because all the protocols we are using are quite variable, and we were actually making the point, okay, this is actually not good, but it's also potential. And I will give you in my talk some ideas why the variability basically also is a kind of a potential. So I would like to present you in this lecture a framework of the target definition and engagement, and I will target two questions. The first is, what do we have in the toolbox? So what kind of simulations we have, and this is a very short introduction, but I will outline why the different methods we're using are different and why they are synergistic. And then what is the problem with the target, and I will use a lot of time more on that. And then once we have defined the target, how can we engage it? I will not talk about the third important step, which is the translation into the clinic and the validation in terms of therapy and plasticity using effects. That's the job you have to do. Starting out with the toolbox, what kind of toolbox we basically have? Do we have a toolbox with a lot of different tools which are quite different, or do we have a toolbox which is quite similar and it's just very small differences with the different methods we are using? And here I just flag up three different methods. Here you could actually add others, and I would just put this new method in as methods which will come in the future and will add to the toolbox being even more different. We have the TMS and the TDCS and the CACS, which are low-intensity current stimulation given through these electrodes, and they have different properties because the TMS is basically acutely inducing a very strong field, which then induces action potentials and actually induces neural activity by its own. This is not the case for TDCS and TACS. It's not discharging neuron itself, but it interacts with the intrinsic excitability activity by shifting the membrane potential. So just to reiterate that here a little bit, we have the TMS and the TECS. Basically, from a biophysical point of view, these two things are also different. For the TECS, the skull is a problem. It's actually the problem to get the current through the skull to the brain. For TMS, which is basically inductive electrical stimulation, the magnetic field is a Trojan horse which brings the current through the skull, through the bone, without any attenuation apart from the distance. So here we have a issue with the skull. The skull is no problem here, but of course once the current enters the brain, then things get also complex with TMS, because the brain tissue and the CSF are not a problem, but they're just shaping the electrical currents, and this is also the case, of course, for the TECS. And again, we have the different intensities which you have to consider. So this is the first part of my talk. They will not be all so short, but this one is and it's just now. And what I wanted to say, yes, the tools are substantially different. There will be new tools on the horizon, which we just have a glimpse of. They target different neural substrates, and it's difficult to translate one finding from one modality to the other. This is just the first start. Now we come to the second part of my talk, which is actually about the target definition. What is a good target? And I will also keep this very generalistic, very kind of, I'm not going into specifics, but I want just to discuss with you what people think might be a good target, and what dimensions we have to think about targets. So this is just a kind of a slide which shows the resolution in space and in time for the different methods we have to stimulate the brain. Here we have some methods which I'm not talking about, but of course, when we're thinking about a target, we have to consider the spatial temporal resolution of our method, because that defines which part of the brain we just stimulate. And we can be very high in the temporal resolution with single part CMS, but we can also be very we have a long-term resolution with the plasticity inducing effect. And of course, in the spatial domain, we are reasonably kind of focal, but we can also be rather unfocal. So I would like to say here that NTBS targets different, and that is, I think, very important. Drugs are not working like that. If you put a drug into the brain, it goes to a receptor or to a molecule. So all the spatial components are more on the molecular level. So in this case, the targets for NTBS are complementary to the drug targets. And I think that gives a very nice kind of synergistic way of inducing, kind of targeting different targets. NTBS targets always target components of brain networks. And think about a target in a neuroscientific way as a neural process that is critical for a brain function or dysfunction, and the therapeutic target would be then a process that actually plays a pathophysiological or pathogenetic role. This is basically a little bit thinking about NTBS and where we're targeting. So I made the point that given the spatial resolution, NTBS targets the brain at the neural systems level. So when you have a specific gene or specific molecule, that's where the drugs basically are working. They're also working on pathways at the cellular level, but then you have the systems level, which consists of the pathways of the microcircuits and so on. But then you're getting, of course, to the behavioral level, where you have the traits, the symptoms, and then the symptoms. So you, normally, if I'm talking about targets, I'm talking about, I'm talking about these targets here. So you target a systems target here, but you have to be careful what it does down on the, because of course it has also a pharmacological target, and it has a pathway target at the cellular level. So here you need basic science to understand this. But also you can say, okay, I'm actually not interested to target a neural system, I want to target the symptom. And that is also fair enough. So you could say, oh, let's target the symptom. But also there you have to think about what you're doing, because when you are targeting a symptom, that might be connected to different traits, and these traits might interact with different system networks. And in the end, you're still targeting a system. You're not targeting the symptom. So you have to make the translation across these levels. And I think people have not a sharp idea about this. It's quite important. And now I'm going just through the target. This is the target which basically was invented at the NIH, early work by Mark Ellis and Pascal Jungs and others, people here, you all know, they target basically the regional cortex accessibility using the TMS and stimulating the motor cortex and kind of recording the motor evoked potential as a readout of accessibility. This has been done for high frequency and low frequency RTMS, but now we have a lot of other protocols which do this. Why should excitability in a way actually change function? And this is just a very, and don't judge me on this, this is just a very simplistic way of how I think about this. Usually excitability should not make a big deal because we are normally in a range which is kind of homostatically regulated. So a shift in excitability might change a reaction time, might change the response rate or whatever, but it should not change behavior in a large scale. But we have diseases where we have a dysfunction in terms of hyper and hypoexcitability. And here the same shift in excitability might actually do a big job because we are in a kind of inverted U and we shift something back to the normal range. So this is basically my rationale why the excitability marker still is a marker we should discuss, and it's still a marker which is used for a lot of new protocols to figure out how it works. It has a problem because we can do this only over the motor cortex. We can, of course, do it also with phosphines over the visual cortex, and we can also look at the CMS-evoked potentials, and I will talk later about it. We can also think about excitability more in terms of the excitation-inhibition balance. And then we can look at pulse TMS, and we can look at spectroscopy, looking at GABA levels. And of course, we can also look at overall regional activity, which is not the same thing like excitability, but is also a regional readout about the activity and the excitability of neurons in a kind of regional way. Here we have all these different methods which have been introduced now. We have a lot of different tools which do presumably a kind of LTB-like or LTD-like change in excitability when applied over the pericentral area. And here are other kind of protocols which have came into play, and they all have been tested here. The problem, as I told you, this rule of thumb has too many exceptions, so it works, but it doesn't work in every case, and we have to look at it. So this is the excitability readout. And the next readout, and I think this is the one which I actually think is most interesting, is the connectivity readout, because this is just kind of the structural connectivity of the visual motor system in the macaque, and you can see that from getting from the visual input to the motor output is all about connectivity. So kind of interfering with this interregional connectivity is also a valuable target. And when you think about the MEP, the motor evoked potential, it's a connectivity measure. You're stimulating the motor cortex, you're looking at the muscle. This is not excitability, it is connectivity. And this is just a prepedeutic slide, which I apologize for that because most of you know this. You can, of course, think about the structural connectivity and do diffusion weight imaging to see what's happening here. We might actually change that with our methods too in terms of long-term reorganization. We have the functional connectivity, which is an interesting marker, which is actually used by people like Michael Fox with the resting stage to actually define targets. But I'm actually more kind of a subscriber for the effective connectivity, which basically makes not only statements about temporal-spatial correlation, but actually about causal interactions. So I think the biomarkers one should go for are biomarkers of mutual causal interaction between areas. And those should be the targets we look. And I will tell you afterwards that this is basically done with all the stimulation. And this is also, again, a very simplistic, but I think maybe useful way of thinking about what we do with stimulation. So we have here a very simplistic network with intrinsic processing, with mutual interactions between subcortical and cortical areas. And if we have a state A, we might actually get in this network into action. But with state B, same stimulation, same coil, same brain, we get this site here stimulated. And if it's TMS, it might be antidromic and autodromic. So this state by stimulation interaction, that shapes the brain, and I think this is the biomarkers we should go for. The last thing is the oscillations. That is another biomarker we are now targeting. And of course, oscillations, when you look at them in terms of phase covariance between areas, it's a connectivity marker. But it's also an excitability marker, which basically looks at fluctuation in excitability and activity at one place. And they, of course, carry information, and I'm not going into detail because you all know that oscillations are relevant, and then the phase-dependent manner shape the processing in the brain. But I want to make 2 points which I think are important when now people hop on the train of targeting this. People say, is it the phase or is it the power? I think it's a stupid question because it's in a way interrelated. Because you only have phase if you have high power. So you cannot target phase with the methods, the algorithms are not working when the power is high. So let's say and this is actually a psychophysics experiment, I'm not going, this is actually a paper worth reading, making the point that the behavioral effects of the phase are only there when you have large amplitudes and high power. If you have small amplitudes, then other frequencies are kind of prevailing, so your average frequency is higher, so your extensibility level is higher. So looking at the amplitude and the phase is not something independent, but is something interrelated. And of course, these measures, which we then use with EEG, they have also correlated at the regional level in terms of neural activity. And we know this by fMRI experiments where EEG is looked at. As you look at the power envelope, they correlate here in this case inversely. The alpha power envelope correlates inversely with the both. So it actually says something about accessibility. And to kind of and then of course, if the state is variable and all these readouts are variable, then it might be not a good idea to do a kind of open loop kind of simulation which disregards the state. What you have to do is you have actually to do a state-informed open loop or closed loop simulation where you read out the state, it might be oscillation, might be power, it might be the ongoing activity or the effective connectivity expression, and then give the simulation and then look at the after effects. But you also might do this adaptive closed loop that you actually use the information and then shape continuously your readouts. I want to make the statement, I just heard it in a recent talk, everyone talks about closed loop. I'm not aware of any closed loop realization. Everyone is doing state-informed open loop. It's still open loop in a way because you're not basically using the information of your effect to kind of modify your simulation. So also, I'm very sharp on nomenclature. Some people say I'm a little bit too sharp on this, but I think it's very important. Don't use these like NIPS or other things. It's really important that the field uses the right nomenclature. And in this case, I think open loop is still the right term. It's just a state-informed open loop when you're targeting a phase. It is not closed loop because the engineer would not consider that a closed loop. We should not leave it the wrong way. So yeah, I would just also to conclude with that part of my talk. So what I would like to say is the targets we are targeting, at least what we do in the humans with our methods, is at the systems level. These targets are all interesting and are all relevant, but they're not independent. So when we target the extensibility, we'll target most likely also a phase or a power measure or a connectivity measure and vice versa. So to have a holistic view about this is very, very important. And then these targets are not fixed, they fluctuate in time. It's the spatial temporal dynamics that unfolds all over the place in the brain. So just going in and simulating without knowing it may be wrong. At least not often. So now I'm shifting to the last part of my talk, which takes most of the time. And this is about, okay, once we have decided us for a target, and each target has limitations, but let's say we have decided for a target, how can we actually make sure that we actually hit a target? And I will only talk about the online confirmation that we actually reach our target. It's not about the offline plasticity effect, which would be a different lecture. So how can we secure it? And then, of course, you have to think about three things. One is where to stimulate. So you have to make sure that actually the current you insert into the brain actually gets to the place you primarily want to target. And the second part is when to stimulate and how, and that is all about the state space. So I will first start with the where to stimulate. And where to stimulate, I think here I would really want to make a case for that you have to do biophysical modeling of the things you're doing. We're doing this quite a lot. Architecture in our center as a Sibner software is developing this, and I would really want to stress that having realistic head models makes a difference. It tells you, gives you an idea, and I can tell you a little bit that you can validate this, but it gives you an idea of where you're stimulating. For instance, here, this is something just came out in New Image Clinical. If you have a stroke lesion with a stimulation, you will get a kind of very strong condensing stimulation close to the cerebrospinal fluid. And we have just seen this in the previous talk. So the liquor, the fluid will actually attract the current, and because it's the weakest way. And this is also relevant for TTCS and TACS. So if we want to have target patients with atrophy or with lesions, we have to do this. I think it's unethical not to do it. And of course, you have other things which I tell you can get insight into. So for TMS, we all say we stimulate the motor cortex, but I tell you, I think it's most likely wrong. So we have said 20 years we have said we stimulate the motor cortex, and that's actually in all the books I have published in articles. I say I stimulate the motor cortex, and I do it transsynaptically, but within the motor cortex. And then by turning the coil around, we get different responses. If you then do the modeling, what you can see is, of course, you get different responses because you're stimulating different sites. But what you also can see is that most of the current is actually produced at the crown, and the motor cortex is in the wall. So you can definitely see at the transition from the crown to the wall, that's where the hotspot is predicted from the models. Is this true? We did a study to look at it. This is just to show you where the motor cortex in humans is. It is in the anterior wall of the central sulcus, and actually also area 3 is not really very superficial. And what we see here at the surface is actually all premotor cortex. This area here, in some subjects, might be the very rostral part of the motor cortex, but it's most likely also premotor cortex. So the question arises: are we actually stimulating the motor cortex? Yes, we do it, but maybe through premotor motor connection. And that we can do this, it actually has been known now for more than 50 years. This is work by Patton and Amazian, where they stimulated over the premotor, but also with the sensory cortex. And what you can see here are I waves. These are the descending waves which descend to the in the corticospinal tract. And when you cool the motor cortex, they are gone, which speaks for that this is a premotor motor or sensory motor activation, and this is happening at very low intensity. So, and this is just other studies from monkey studies, where you can see very short-lasting effects, very short-latency effects in the cortex, and actually kind of like an MEP sign field pattern, just in animal in the motor cortex in these monkeys. So what we did is we did that what we call a shape-based mapping approach. So it's a very primitive thing. We use the shape of the sulcus. We use the neuronavigation. We use information we have by structural imaging. We defined 7 points which we put over the central sulcus, and then we shifted this line more and more in front. And then we look where the hotspot is. And then what you can see is that the hotspot is basically in some subjects more in the premotor cortex, more rostral, and more lateral, while in the others it's more dorsal and more close to the central sulcus. And then we said, okay, let's split that in half and call one of them the motor cortex subjects, which I think maybe are the core premotor ones, but let's call them motor cortex subjects, and the others the premotor subjects. And what you then can also see is that they basically have different latencies in cortico-muscular latencies. So those who are more rostral, they have a longer latency. So it takes longer to arrive to the muscle. We did in these people also fMRI, and we used the tapping task with the left hand, because we stimulated the right motor cortex, and we looked at the peak while they tapped with the index finger and the little finger. And what we saw is that there is, of course, a difference between the index and the middle finger with the TMS, and also with the fMRI. You see the peaks are much closer, but they're still significant. But if you inform by your latency values, you can actually see that there is a dissociation in the spatial representation also in fMRI. So those premotor subjects have a more rostral representation than the motor cortex. If you then go back to the SIMLIPS and look at the field and compare the two groups just based on the latency and then the position, then you figure out that those who have a stronger activity here in this rostral area, BA4A, that they have actually a higher field here. The field in the ground is matched between the subjects. So these people have a higher field here. So those where you actually hit the motor cortex more closely, they have the shorter latency. And when you then go back to the fMRI data, then you can see that in the motor cortex, the fMRI data it's the same. But in the premotor mass, you can see that only the premotor subjects have a stronger premotor activation. So this makes actually they also have a better competence in doing this more regular. So they actually these two groups are different in terms of behavior, in terms of their fMRI activity, their latency, and the induced field in the brain. And actually there are also structural changes, which I'm not going into. So this means we have two different groups which we most likely target differently, and one is definitely premotor, and the other one is maybe rostral motor or caudal premotor, we don't know. But where the pyramidal cells are, most of them in the ball, that's not what we're targeting. So it's actually a premotor motor, or maybe a sensory motor stimulation. So that's what I think what really happens. Here is the bulk of the motor cortex, and we have two different groups of people, some who have a spot more here, the other more here, but it's most likely that even this area is not a real clear motor area. So I think it's basically, we always have done a premotor stimulation. It's just the kind of location. But now bringing this to the TDCS, I think it's even more important doing this modeling. This is the classical montage that has been used, and endless papers have been done about stimulation over the motor cortex. But most of these stimulations very clearly show we have stimulate a lot of areas, very speckled and very inhomogeneous, but it's more in the prefrontal area and the premotor area. The motor cortex actually is not really stimulated here. And that would also speak to that this is sufficient to boost the motor cortex accessibility. If you then use a ring electrode, you can get closer, and if you use this pseudo-monopolar arrangement, you also get closer. People now use 5 or 6 electrodes and have again labeled a very kind of claiming high definition nomenclature, which I also think should be forbidden. This is not high definition. It is actually because the brain is evident, the current is smooth. And there is a limit how high definition you can be. It's the brain is smoothing it. It will never be high definition. This is just a marketing thing. Should call it a multi-electrode tax or a pseudo-monopolar or a ring or a dipolar. Stay to the description, but don't make claims. And you can see that this is different. And this is not as focal as TMS, maybe less focal. I think TMS is still much more focal than this. So I think these things are not helpful only for the vendors to get the machines sold. The classical bipolar montage, as I told you, is basically not hitting the target. It actually hits a lot of different areas in between. A quarter of the brain might be stimulated in a speckled fashion, some in the depth of the cycle, some in the superficial way. So this is terrible for cognitive neuroscience, because cognitive neuroscience has used TDCS to make claims about focal localization, which I think are not valid, because this stimulation is not focal. We did a very early PET study where we looked at the blood flow changes with this montage, and we were at that time puzzled because we found these kinds of speckled way if we look at anodal versus cathodal during this arrangement. But now we are more confident that this was a real finding. It actually reflects some of the speckled effects here, but at that time we could not explain it. So I think there's a face value here which is quite striking. Then, of course, if you use these bipolar montages, and this Axel Zilcher's work, it makes a difference how these electrodes are made, or where the electrode comes in, how homogeneous the conductivity is, and that makes the travel lot of a difference. So if you get this wrong, you don't need to talk about physiology, because you just biophysically getting different targets. And now the last part is, and this is just another iteration of that the field is ignoring the biophysics. This is there are a series of papers out which have used tax in an in-phase, anti-phase way, cognitive neuroscience, to figure out that the phase processing is relevant. And I was kind of puzzled because when I saw the montages, I was kind of really thinking this makes no sense. And then I went to Axel and said, could you do this kind of simulations? And we have just published this in Neuroimage, giving you all the papers that have been published so far have not got it right, and we show explicitly what they actually have stimulated. This is just one example here. This is one of the first papers published in Current Biology, where you have these 3 electrodes and in-phase and anti-phase. But this is called a reference electrode. But of course, the current doesn't label itself as a reference. The current goes out here at a double intensity than here in the in-phase condition, because it's just one electrode. The current goes out through it, so you have a double intensity here. But what the idea was to actually target selectively these 2 areas. And then you can see in different conditions, you're getting completely different spatial patterns, and they vary over time. So of course you get different effects, but this has nothing to do with anti-phase. I think all these papers should have a kind of erratum actually to comment that this is basically wrong. And I'm quite sure a lot of people out there are using these methods now and get their PhD kind of mismatch. So this is very, very important. And what you can do here is you can do it properly if you use 4 channels, and you can actually look at interhemispheric phase coupling. But you should not use the 2-channel stimulator. Some people actually then kind of couple the small electrode here with the ring electrode here, and vice versa. Then you get terrible different effects. So if you use a 4-channel ring kind of arrangement, you're actually able to get the phase right. And you can also do this intra-hemispherically. Here it is shown in this table, so you can actually do in-phase and anti-phase comparison. So it's possible, but you have to do biophysics. So I just want to kind of summarize this part, which is more controversial, and I'm looking forward to your comments. I would say not all brain targets are created equal. The brain has a bias where the current goes. For TMS, it's the surface of the brain, so it's very difficult to actually physically get the depth. For instance, the depth of the cycle is very difficult to target. Therefore, I think a whole brain connectome's resting state activity modeling pathway is important, but you need to implement the bias here too. Biophysics should not be ignored, and this is true for TMS, for bipolar TTCS, but also for other things. Phrases like deep TMS or high definition TTCS should be banned because they are wrong. It's not deep TMS, it's high volume TMS. You can do this with the ground coil too. I have a lot of discussions with people using it. I was just at a meeting in Munich, and there were 10 posters with this thing, and it's basically the thesis using it. I think we should do something about it because it's wrong. It's not like deep brain stimulation, and people think about DBS when they hear deep TMS, and this is misleading. The strongest current is at the surface, and you might hit the depth, but at a very low intensity. So this is very, very important, and I think the field should stood up because it brings us in a problem because people who know about it, they will not take us seriously. Okay, focality is important, so we have this obsession to be more and more focal. I think this is fine for cognitive neuroscience, and I showed you that the bipolar TTCS was the wrong way to do it. But large volume NTBS might actually be important. Maybe in patients, this is actually a good thing. So I think we should be open-minded about it. I'm not saying focality is important, it depends on what you do. And the last thing, which I have not talked about, but I will talk now, is about the periphery. The periphery is always in. When you do the stimulation, your trigeminal nerve, your vestibular organs, your retina, they are very happy about current because that's the way they get the senses right. So therefore, it's very important that you also have this in. So if you do the modeling and you're targeting, think about the peripheral side. Having said that, I will now shift to the when and to the how, which I think is even more important, but let's have a look. I already told you about the TMS, and that the TMS over the motor cortex is the classical readout. So here we have a readout about the excitability, but also about the connectivity, and it's easy to use. But it's, of course, very limited, and as I told you, I think it's not the motor cortex we're stimulating, but the premotor cortex. But that would just add another level of complexity. Another way of looking that we actually get our target right would be a functional readout at the behavioral level, and this is completely valid. And I just give you this example here about the kind of short visual blindness, which you can induce by optical TMS. And of course, once your intensity is high enough to get visual blindness, you most likely have stimulated the optical cortex. So this is a perfect valid way of figuring out the dose. But otherwise, it's quite difficult to actually make sure that the intensity we use is actually sufficient to get the target right. So we need brain mapping methods to help us. And this is just a way to kind of conceptualize our brain mapping methods, and I might have forgot some of them. But the point I wanted to make is the area they are covering is basically the area we're targeting with TMS, TTCS, and THCS. So we just need to be smart and looking at these methods and bringing them together with the imaging, with the simulation, and then we get actually quite interesting results. We can actually also look at a very global effect with a very local TMS and see how it spreads out. So I think that we actually have a good toolbox here of methods which we can use to target. One new method is a relatively new method is the EEG, the TMS evoked potentials, because now you're no longer limited to the motor cortex, but you can spread out to the other areas. And there has been a lot of work done in sleep and in wakefulness over different areas, and you get these responses which are kind of lasting 300, 400 milliseconds, and they differ between areas. But do they tell us really about target engagement, so about the very acute thing which happens when the TMS hits the brain? There's one study by Casarotto where they basically used from 20 to 50 or 60 milliseconds the responses, and then made a kind of cumulative marker, and they called this the immediate local response. I would say if you really want to know what's going on, you have to be there already in the first 5 to 8 milliseconds, because that's where the TMS actually excites the neurons, and there we have our artifact. So that is still a challenge. So, but I would actually discard this as an immediate target engagement, and I'll tell you later it's most likely isn't. But that would be actually quite a nice thing. If we can get the artifacts, handle the artifacts, we can actually do this with the tabs. This is a study which I would like to, which is not published, which is relevant for a lot of people now using the tabs, because of course with the magnetic stimulation we have a lot of peripheral input. We have somatosensory input and we have auditory input. And what we can, but it has been actually tried. There are some studies out who have looked at how much the peripheral input contributes to the TEP. But we were not so happy with them, so we tried to do this in a proper study where we had a real simulation over the mediofrontal and the medioparietal kind of targets, and we did this with the coil turned around and different coil orientation, because that should give also different readouts, and then we used a kind of placebo stimulation with a wooden block in between, but with the bipolar electrostimulation here at the skin to mimic the stimulation. What we found with this method, which was not a perfect match, but was close to what you feel when you have, and of course noise masking was done. So even if it was not perfectly matched, we get very, very similar curves using the global mean field power, but also the topper plot with the conditions. TMS orthogonal, tangential, and sham. You can see that the temporal modulations are very, very similar, and the topology is very, very similar. If you then look at similarity, so you systematically look how similar things are. So here we correlated for each point in time all electrodes and all values at a given point for the three conditions, for the two conditions, and then made a kind of average correlation matrix. You see they are highly, highly correlated. So you can see this here. Basically, it goes up for this TMS tangential versus sham, or TMS orthogonal versus sham. The blue and the red are very close to the green. So that means most of the information we get with the TEP is actually perfectly generated, and I think you need to validate this with intracranial recordings and really figure out what you're really measuring and what is really neural. And I hope some people here in the room soon start with this, because I think this is really important. So this is actually a readout which is also dirty because we have the sensory input, but a lot of the markers are based on consciousness and so on. They are based on this, and I would say that these markers would also work with our sensory stimulation as good as with the transcranial stimulation, because it's a sensory signal. We can use the fMRI as another method, and the fMRI is of course not good in the time resolution, so we will not get really the milliseconds, the first milliseconds right, but we have the whole brain resolution. And this is work done by Sven Bestmann, which I think still is very important similar work, where he actually stimulated the premotor cortex with bursts, and then showed a target engagement in the premotor cortex, which was less than during a voluntary contraction, but actually indicating that there was some activity. What in this study was beautiful, but kind of not really emphasized a lot, is that there was actually a very nice deactivation on the other side in the sensory motor kind of cortex. So we know that we have this transcallosal inhibition with the paired pulse stimulation, so cortical protocol, and you get this inhibition here. And you could see this, you can see this very nice increase in the activity in the right sensory motor cortex, stimulating the left motor cortex, which is not there when you have a movement. But it's actually already there when you stimulate at 90% active motor threshold, where you not get where you don't get the inhibition. So the fMRI readout is actually much more sensitive to figure out that there is a kind of a decrease in activity, presumably due to inhibition. So this is a very powerful readout for looking at local effects and global effects, but with a poor time resolution. In the last 5 5 minutes of my talk, I would like to now switch to the state dependency, because it's not enough to just bang the brain and zap it and then look at the reader at what happens, but it's, because things are fluctuating, these methods have to be used in a state-dependent fashion. And this is not new. We knew that from the TMS in the first place when we used it clinically, when we stimulate the motor cortex, premotor motor complex, and we do a pre-contraction, same intensity, our MEP just becomes larger. And it actually becomes mostly larger, not because there are more neurons stimulated here, because the summing up at the spinal cord gets more efficient. So the spread out through the system gets better. And this is massive. It's from a null response closely to a huge response. So this is it is nonlinear because at 10% motor contraction is there, and 25% it's the same. Now we discovered the nonlinearity. We knew this already 25 years in the clinics. So the response is nonlinear and it's state dependent, and we have to figure that out with our imaging methods. But it's not new. People have known that before. Just want to make that clear. And of course, if you look here at the TMS, when you give it during different power states, then for the visual cortex, the probability to evoke phosphanes is kind of dependent of the alpha power, and of course the TMS over the motor cortex is kind of also dependent. The amplitude of the MEP also depends on the power of the alpha. So the oscillatory readout is also kind of modulating this connectivity accessibility readout. This is a study we did in sleep. It was the first study where actually the TMS was given in an informed open loop fashion during the up and the down state of a slow wave while people were sleeping. And you can see when you apply in the up or in the down state, this is actually the response during wakefulness. You can see responses are massive. Basically, we're evoking a slow wave when you give a TMS pass. But you can see that the amplitude is different whether you're in the up and the down state. And for the early responses, it's quite focal. For the late responses, it's quite unfocal. And what you also can see here is that this state dependency not only affects the slow wave that you are inducing, but it also kind of induces a neural motif which is embedded in the system, because we know that the spindles are actually clustered around this phase of the slow wave. And you can see in the up state, you get a more intense spindle clustering than in the down state. So you're not only evoking one oscillation, but you're actually invoking a motif the brain actually is generating in this particular state. And then, of course, led us to push this towards the wakefulness. So in the last years, we basically worked on a system which we tried to make open source, where we have this new EEG amplifier, which gives us a high recording sample rate, and we record the online EEG, and then using a Python script, trigger in the face. And what our first experiment was here was to target the alpha in the pericentral region using an approach where we define a local source, a local radial source, and then target this kind of monitor the source activity online, and then target in the up or down phase, but also in the ascending and descending phase. These are subjects, they were not selected. Some people have a bad neurism, some have a good. We just took them all in, and it basically works in everybody, but it works best in those who have a neurism which is kind of prominent. And then you can see here that we basically, here we estimated, from here we estimated the response using a Wallace transform, and you can see that we are in the up and the down, and here basically we have the similar thing for the different phases. But you can also see it's noisy, and you can also see that the alpha activity is waxing and waning. So alpha is basically then decaying. So it's not as easy as we expected. But we got the phase right, but in 3 experiments so far, we were not able to kind of really find a consistent change in the MEP amplitude. This is the data from the different target phases which we used: 0, 90, 180, and 270. This is the amplitude absolute values for the single subject. For the monophasic in the first 3 blocks and block 4 and 6, because we were wondering whether something was there in the beginning but not in the end. And this is just the mean normalized way, and this is the mean data across all blocks. As you can see, there is not a consistent pattern. So far we were not able to kind of figure this out. Our hypothesis is when we select only those with a very high repower and looking only at those with a very high amplitude while we're targeting, we see a trend towards this. So this only works when people actually expressing it at high power. And again, this basically kind of confirms my suggestion that power and phase are not independent, but the phase is most relevant when we have a high power. The very last experiments which I want to show about phase dependency, and I think that's a very beautiful way of doing it, would be that you with two coils try to target a cortical-cortical connectivity and then use the motor cortex as a readout. So this is a well-established paradigm. We use here a method, so here basically the state is induced by us. So it's not the brain inducing the state, but by using a conditioning pulse, we ping the brain, and this gives us a state, and so we don't care about state, we insert it, and that's maybe a better way strategically to do it, and then we look what happens on the motor evoked potential. The trick here was that we gave a premotor pulse, but the premotor pulse was actually given after the motor cortex stimulation, which sounds a little odd because the conditioning pulse usually is before. But since we know that the E waves are leaving the motor cortex 6-8 milliseconds later, we thought that this conditioning pulse has plenty of time to reach the late I waves. And therefore we thought there should be an I wave kind of facilitation, which actually was the case. So at rest, you have this I wave facilitation. It's very valuable for subjects, again, most likely because of the difference in anatomy, but you have this I wave facilitation by giving a premotor pulse after the motor cortex pulse. When you then do an activation study, like we do in fMRI, but I call it the single pathway activation. So it's a single voxel, it's just that pathway we're looking at. And then you do a motor task where you ask subjects to prepare to do a movement depending on a cue. So here you have a cue that triggers this finger movement, and here you have a cue triggering this finger movement, and then you give the signal or the pad pulse in the reaction time period. Then you would expect, depending on which cue comes, that the premotor motor activity will increase for the pathway that basically should be activated. And that's what happens here. So if the cue triggers the movement with the left finger, so a left premotor, right motor cortex pathway, then nothing happens. Actually, there was no inhibition, no hesitation there. But when it triggered the right movement, actually at 100 milliseconds, inhibition was completely gone. Error bars were low. So here basically you have a state which is well defined. But then in those subjects who actually did the response, then the facilitation came up again. So this channel is there at rest, but also during task selection, with a superb temporal spatial resolution, which you cannot meet with EG or with fMRI. So with that, I would like to make a summary about this part of my talk. So I think target engagement is something very important that should be assessed, and I'm talking about the online target engagement. We need to know what we actually do to the brain while we're stimulating it. It is a challenge for TACS and TDS due to chart effects, but the same is true for TMS. So the acute effects are very difficult to target, and there are a lot of methodological challenges, and I think it's still not really possible to do this. And with fMRI, we'll have mixtures of early and late effects. What is easy to do is to look at the network effects, so the spreading out of the stimulation to the network, that we can measure, and this provides a really important mechanistic insight. The intrinsic states can be traced by EG-TMS or externally imposed by text or by cortical paths. So these are two ways of playing with the state dependency, either waiting that the brain creates the state or doing yourself insert the state into the brain, right? But very, very important, the brain stimulation also excites the peripheral system. The TEPs are massively confounded by that, and this needs to be considered in consciousness studies and other studies using readouts. Of course, also in MRI, we always see auditory activation where we could give TMS, but it's more localized, so we can better disentangle it. And with that, I would just like to summarize the main point. We have to define the target. We have to know that the stimulation is engaging the target. We can then refine the TPS and then go through this loop over and over again. And this iterative cycle is the one which is important at the human side. This has to be supplemented by basic research, but this is the way how we can actually solve it, not having animal facilities and working with humans. With that, that, I would just like to thank you for the attention and that no earthquake is now arriving. Questions? Please.
Well, thank you for the talk. It was amazing. I will go to something quite specific. You mentioned that it's impossible to estimate the state change immediately after the pulse. Now, with every pulse, you are depolarizing a whole bunch of networks, and everything is under the coil, right? Then the networks will start to communicate and reestablish the local homeostasis, electrical homeostasis, right? So I suppose these systems will come back online one at a time or in parallel, but with different phases.
Yes.
When would you stop looking at a meaningful change? In Vermein's paper, for instance, he relates that change 500 milliseconds after the pulse. Is this too long to have any?
So your question is, when does the acute impact of the stimulation kind of really stop at the site of stimulation, right?
Or when the complexity explodes and it's not meaningful anymore to look at the effect.
Yes, okay, but I think there's two things we have to kind of differentiate. The one is exactly the hypersynchronous stimulation that we do with the TMS pulse, which in a way is a spike wave complex. So what you do is you stimulate a large part of EPSCs and you generate EPSCs at short and kind of GABA A and GABA B, and they will give all these effects which we have. And this you want to kind of dissociate. You can do this with pulse protocols a little bit, but that's the very acute impact of the TMS on the target. And then, of course, then is spreading transsynaptically and sometimes also antichromically, and then we are retrograde actions actually even also transsynaptically out to other areas, and then that does a kind of network echo, and that can come back to the cortex. And of course, and so these are the two different things. And what I think is important to dissociate between these two things. And we have no clue what actually happens locally when with the methods we have at the moment to dissociate this from the rest. And I think this happens within the first 5 milliseconds. So I think that's 5 to 8 milliseconds. That's because then the reiteration it might not be happening. We know that the transcallosal inhibition is 8 milliseconds, so it can travel actually quite fast. So I think we should not go at 10, 20 milliseconds. But all the other things, they are kind of a combination of the local impact and then kind of recursive activity to that area. And I think it doesn't make sense to say when that stops. If you from a clinical point of view, if you have a TMS and you get a MEP, often it has not been studied very systematically. Often the first MEP is much higher, and then a lot of MEPs coming, and they're variable, but they're much slower. So there might be some long-lasting effects with the first hit, which we have not really systematically looked at. So I would be actually very cautious to say when does this stop, because what you do is highly unphysiological and it's very close to a spike wave complex.
So one requirement for targets that you haven't mentioned is relevant to some clinical or behavioral outcomes. And we, for many years, chose the target of the MEP. We said if you could modulate this up or down, then fine, you're modulating mood or something like that. So what do we have now that's relevant?
Okay, so the question was, we have these electrophysiological kind of targets. What do they mean in terms of behavior or function or dysfunction and pathophysiology? I would say that I personally think I completely share the point. In the last part of my talk, basically, this kind of cortical path experiment actually showed something about behavior there, because in a way it was irrelevant for action selection. So this cortical pathway was relevant for this. But I agree, it's a challenge to translate these things into function. For the oscillations, we're actually in a much better position because we have a lot of electrophysiology which tells us how these oscillatory patterns are at cortical interactions, how they are related to mechanistically to behavior. It's very difficult for me to find out to say why should a cortical spinal motor readout be a general marker for depression. I cannot see this. but it is a marker for stroke recovery, or so I would say this pathway is very important for independent finger movements, and within that framework, it's skill movement. Within that framework, this is a good readout and a good marker.
Has anything been validated?
No, I think I'm personally, that was the last part of the talk which I left, because I didn't want to be too pessimistic. I was actually quite critical also for the first two steps. But I would say the field has a challenge to define markers that are relevant. There was actually a nice talk here some months ago where basically the starting from a frontal polar target and effective connectivity and to try to bridge that. And so there are attempts, but whether they are really validated and then brought into a kind of controlled blinded study design and then confirmed, no. Eric?
No. Yeah, I think you have been too generous to TDCS, especially the modeling, which all the modeling you showed took into account only circumferential current flow, as far as I can tell. And if you begin to look at radial flow, it is a really complex and chaotic picture. And I don't think the so-called focal concept has been validated biologically. And we couldn't get it to modulate the excitability of the motor cortex. And I think it's another case where it's much more complex than it looks. So I would support everything you said.
And this is extreme. I'm very cautious what to say. I can tell you that we don't do TDCS research in our lab, and we haven't done this in the last 10 years.
Right.
But that's my personal take on it. I only use methods which I believe in. But I think that TDCS might have some value in terms of, if you think about the TMS, it gives an awful strong current very, very shortly. But if you look over time, how much current actually goes into the brain over that long time, right? It's actually quite a lot of current. But what I would say, it's very complex. I think it's useless for cognitive neuroscience, and all these papers who make this kind of local focal kind of claim, they have to be thought of. But I think the jury is still out here. But it is complex. It is even more complex than for the others. But the TAX has a huge advantage. It might be very, very unfocal, but your brain oscillates and engages at the oscillations in the brain. They will just decide on which TAX protocol to hop on. So the brain will localize it.
There is a biomarker of target engagement.
Sorry?
There is a biomarker of target engagement.
Exactly. Yeah, but there's also a rhythm in the brain. There's a biophysical kind of language in the brain for this.
Yeah, that's the target.
But for TDCS, we have it actually for the slow potentials. So maybe people should think about slow potentials, readiness potentials. Nobody has done that so far. But I think the jury is still out.
So thank you for this excellent talk in which you demonstrate the qualities of rigorous science using non-invasive brain stimulation. And I just want to reiterate a few of your key points. The field has been ignoring the biophysics and relying on intuition, and this has been misleading. Also, there are ancillary effects of the stimulation that contaminate our supposed biomarkers of TMS evoked potentials and TMS fMRI that are incompletely understood in the literature. And another point that you made was that deep TMS and HD for high def are misnomers, and incorrect utilization of language and terms can muddy the field. And you made the penalizing challenge that we should do something about this.
Yes.
You mentioned last time you were here there was an earthquake in D.C. So how are you going to move the NIBS Earth to do? What do you mean by do something about this?
I think one could, for instance, do an editorial in the relevant journals. We should think about that here, but also in clinical physiology. So the editors should just make a disclaimer. If I, as a referee, kind of alert people to that, I get the kind of very upset responses about being biased and being so I think it's really something the field should challenge and say. And of course, as you say, we see this in politics. Words are creating facts, but the facts are not there. And I think that the thing is, and we're doing the same thing, and so I think this should and we should be sharp on this. And I judge my colleagues and my collaborators on how they use their language, because the language tells how sharp your mind is. So I think we should do something about it, maybe sit together and make editorials and other things, because this is just like we have these echo effects in a society where people just brainlessly reiterate these things, but there is commercial interest behind it, which makes it even worse.
One quick question, more specifically, we're very interested in TEPs, although you have really discouraged that in my mind. But
yeah, I have discouraged it.
We're aware of all the superficial effects on the TEP. So in ERP scientists normally would do something like do a subtraction of, the TMS plus the effects and then just the effects alone, which you've done. Have you thought about, does that work?
I think I think for any study you need to do the peripheral control. I'm not referring to the two papers which have not really addressed it properly and say that we can neglect it. I think that the best way of doing it is first to make the control condition. And I think this would be a requirement for publication, which is not at the moment. The second point would be then, how do we deal with it? Is the structure the right way? And I think for some stuff it is. But for instance, for past experiments, it is not. Because the past experiments are fundamentally flawed. Why are they flawed? Because if you do the pulse with the TMS, we have a hierarchical system. It goes, you have two pulses, and it goes from the motor cortex to the muscle. But in the pulse TES, the first pulse can condition the second, but the second can condition the late responses of the first. And that is this is non-linear. So subtraction is just misleading. It's done. It's misleading. Pulse experiments with PEPs should be abundant. But this is, now you have said this, and I didn't want to put this in my talk, because there is a problem with the fundamental problem that these two pulses, the brain is echoing ping-pong, so the second pulse is conditioning the late responses of the first. And this has not been addressed in the field. So these readouts which we have, I think it's called N100 and so on, they are not valid.
Great. Well, on that note, thank you very much.